Welcome to the discussion on differential staining. What is differential staining? What are the different types of differential staining and how differential staining can be used in the medical diagnosis of diseases. In previous lecture, we have seen that through simple staining, with the help of a single dye, we can study the morphology and arrangement of bacterial cells. Now, in case of differential staining, we use two or more stains to differentiate between the different bacterial groups or the different bacterial cell parts. The different stains which we use in differential staining is based on the chemical properties of bacterial cell wall or the cell components. Now, in the case of differential staining, we generally use a first stain which is known as primary stain. The primary stain generally imparts color to all bacterial cells. The primary stain can be removed by a decolorizing agent. Now, this agent generally removes the primary stain or it may not remove the primary stain. If the primary stain is removed, then the cells become decolorized. As a result, we won't be able to see them under the microscope. So, in that case, we go for a counter stain which imparts or gives color to the decolorized bacterial cells. So, in this way, on the basis of chemical properties of bacterial cell, we choose what kind of stains we want to use. So, accordingly, the function of differential staining can be divided into two main parts. One, it can be used to study the bacteria groups, or we can divide the wide spectrum of bacteria into groups based on differential staining. Like we can divide the bacteria into gram-positive or gram-negative bacteria based on gram staining. Again, gram staining is a differential staining. Similarly, with the help of acid-fast staining, we can divide the bacteria into acid-fast group and non-acid-fast group. The other function is it helps in visualization of structures, like it allows us or helps us to study the flagella, capsule, endospore, nuclear. I have talked about that the differential staining helps in medical diagnosis of diseases. We know that certain pathogenic bacteria like Klebsiella pneumoniae, which causes pneumonia in human beings, they contain capsule. So, if your clinical samples contains Klebsiella and we perform the differential staining with the clinical samples, and if capsule is seen, this means it helps in identification of the pathogenic group of bacteria. Gram stain procedure was first discovered or invented by Hans Christian Gram. He was a microbiologist and he developed this technique while working in the City Hospital of Berlin with pathogenic samples. The function of gram stain procedure is that it helps us in the classification and identification of microorganisms. How it helps? The whole or the purpose of gram stain method is based on the chemical characteristics of bacterial cell wall. Now, if we look at this diagram of gram-positive and gram-negative cell wall, we find a difference. If this is the cell membrane, we can see a peptidoglycan layer outside the cell membrane, and the thickness of the peptidoglycan layer in gram-positive bacteria is greater than we see it in gram-negative bacteria. Another important feature of gram-negative bacteria is that it contains an outer membrane outside the peptidoglycan layer. Now, let us go to the chemical composition of cell wall. The gram-positive bacterial cell wall are generally rich in proteins and mucopeptides. So, when they are subjected to alcohol treatment, it develops or it rather dehydrates the bacterial cell wall, and thus there is a closure of pores, which does not allow the crystal violet iodine complex to be lost from the bacterial cell wall in case of gram-positive bacteria. In the case of gram-negative bacteria, the outer membrane which is present contains huge amount of lipids, and we all know that lipids are soluble in alcohol. So, it results in formation of large pores through which the crystal violet iodine complex is lost from the cell wall. This difference in the bacterial cell wall helps in the color development. We require 1 gram-positive culture, 1 gram-negative. In this case, Escherichia coli and Staphylococcus aureus. We require clean, grease-free slides, inoculating loop, spirit lamp, blotting paper to remove the excess water, and a microscope. Reagents required: crystal violet, grams iodine, 95% ethanol, safranin. First, we make smears of Staphylococcus aureus and E. coli on 2 separate clean grease-free slides. We allow the smear to become air dry, normal microbiological practice, and then we heat fix the smear. After the smear is heat fixed, it is flooded with primary stain, which in this case is crystal violet, kept for 30 seconds. The excess stain is washed off with distilled water. Then grams iodine solution is added to the slide and kept for 30 seconds.
Ma'am, why is iodine solution used?
Iodine in this case or in this kind of staining procedure acts as a mordant. It helps in the fixation of the stain to the biological tissues, as a result of which it is not easily decolorized while using water or alcohol. So in this case, iodine, it reacts or complexes with crystal violet, forms crystal violet iodine complex and binds to the biological tissues. Now, after iodine is kept for 30 seconds, it is washed with 95% ethyl alcohol. Till that point, until no color is given out from the smear. Then it is washed with distilled water, and then the smear is counterstained with safranin and kept for 30 seconds. After 30 seconds, the excess safranin is washed with distilled water. The smear is air dried and seen or visualized under the compound microscope. If the organism is gram-positive in nature, then it will retain the primary stain, that is crystal violet iodine complex, and hence it will appear purple in color. If the organism is gram-negative, since it contains large amount of lipids in the cell wall, so as a result, the crystal violet iodine complex will be lost from the cell wall and it will be counterstained by safranin. Hence, the organism will appear red under microscope. So, in this way, we can see the E. coli, which is a gram-negative organism, has taken up safranin, and hence it is appearing red in color when seen under compound microscope. While in case of Staphylococcus aureus, since it is a gram-positive organism, it will appear purple in color, and so we can say this Staphylococcus aureus is a gram-positive organism. As well as it helps us to identify the morphology of bacteria. For example, we can say that E. coli are small rods, while Staphylococcus aureus is round-shaped in nature. Now, what are the precautions which we need to follow during the experimental procedure? The smear which is drawn should always be thin and uniform. During the heat fixation step, overheating of the smear should be avoided. The third point is over-decolorization of the smear should not be done. So, if we follow these precautions, then we will be able to get good results during gram staining method. Let us move to another example of differential staining, that is acid-fast staining. Now, this staining is exclusively used to identify Mycobacterium. Species of Mycobacterium, they are pathogenic. So, it helps us to identify the pathogenic Mycobacterium in clinical samples through acid-fast staining. Now, in acid-fast staining, generally we use carbylfuscin. Now, carbylfuscin is made soluble in 5% phenol. The reason behind this is that the carbylfuscin is insoluble in water or alcohol. Now, generally the cell walls of Mycobacterium contains a lipid which is known as mycolic acids. The presence of mycolic acids in these Mycobacterium makes the staining of Mycobacterium through other processes very difficult. As a result, acid-fast is exclusively used to identify.
Ma'am, why is carbylfuscin used as a primary stain in acid-fast staining, not other primary stains?
Mycolic acid is generally soluble in phenol. Carbylfuscin is made soluble in phenol. So, the carbylfuscin will automatically bind with the mycolic acids in the cell wall of bacteria. As a result, in this case, we can say that phenol is acting as a mordant. We require Mycobacterium smegmatis culture and another organism which is non-acid fast, like in this case we have taken Staphylococcus aureus. The staining reagents, which includes carbylfuscin, methylene blue, and a decolorizing agent which is acid ethanol. Apart from this, we require clean grease-free glass slides, inoculating loop, spirit lamp, hot bath, or we require a hot plate, then distilled water and a microscope. We make a mixture of Mycobacterium and Staphylococcus aureus culture on a clean grease-free slide. We allow the smear to become air dry, heat fix the smear, and then flood it with carbylfuscin on a hot plate. Heating is applied during the application of primary stain, in this case during the application of carbylfuscin, to facilitate the penetration of primary stain into the cytoplasm of bacterial cells. Then we keep it for 3 to 5 minutes on that hot plate. Then we cool the slides, wash it with distilled water to remove the stain, and then we apply the decolorizing agent which is acid ethanol and wash it till there is a faint pink coloration of the smear. And then again we wash it with distilled water to remove the decolorizer, and finally we counterstain it with methylene blue and keep it for 10 to 30 seconds. Or in certain cases, it is kept a little longer for 1 to 2 minutes. And then we wash the counter stain with distilled water, air dry the smear and see it under the microscope, under the oil immersion lens. Now, what will be the observation? The observation is those organisms which have mycolic acids in their cell wall will retain the primary color, that in this case is carbylfuchsin. And once you cool down the organism, then carbylfuchsin is retained in the cell wall of Mycobacterium. And hence it will not be lost during the decolorization step. That's why it is known as acid fast. It is able to resist the decolorization by acids also. However, the organisms which are lacking mycolic acids in their cell wall will be easily decolorized by acid methanol, and they will take up the counter stain methylene blue and appear blue in color. In this diagram, we can see that there are two different organisms. One is blue colored, another is red colored. So, the red colored is Mycobacterium smegmatis. Why? Because it has written the primary color, while the blue color is that of Staphylococcus aureus, because it is counter-stained by methylene blue. So, in this way, acid-fast technique or staining was developed first by two German doctors, Zeil and Nielsen. And accordingly, this method is also known as Zeil-Nielsen method and is used exclusively for Mycobacterium staining. Since we are applying heat here, so one thing has to be kept in mind, that during the heating procedure, the stain should not become dried. You have to keep on adding the primary stain at regular intervals. The second point is, the stain should not boil. It should be to the point of steaming, but the stain should not boil at any case. And over decolorization of the stain is not required. Now, let us move to those kind of differential staining which are used for visualization of bacterial cell parts. First, we will talk about a bit about endospores, and then we will see how differential staining can be used for visualization of endospores. Endospores are formed by certain anaerobic genera like Clostridium or aerobic genus like Bacillus. Now, these endospores are generally formed inside the vegetative cells under adverse environmental conditions. Now, what kind of conditions? Like when there is a depletion of nutrients in the culture medium, or when there is a adverse physical conditions like excess heat, excess chemicals, radiations. It leads to the formation of spores called endospores. Now, endospores cannot be stained by regular staining procedures because they contain impervious spore coats which are impermeable or resistant to stains and dyes. As a result, normal stains and dyes are not able to penetrate the endospores. Now, in this case, we use the primary stain malachite green. Now, as I have told you, these spores contain impervious spore coats. So, you need such a stain which will penetrate across this barrier of impervious spore coats and enter the cytoplasm of spores. When we are using malachite green and applying heat, those cells which contain endospores will be totally stained. Even the vegetative cells without the endospores will be totally stained with the primary stain. Now, when we are using water as a decolorizer, then only the endospores will retain the primary stain, while the rest of the vegetative cells will lose the malachite green.
And why is water used as a decolorizing agent in endospore staining, not acid or acid alcohol?
Well, now malachite green is made soluble in water.
Okay.
So when you use a solution which is made soluble in water, so you have to choose decolorizer as water only, because the primary stain is soluble in water. Okay. So when we use water as a decolorizer agent, so what will happen? These spores will only retain the color. Why? Because the stain becomes locked inside the spores. As a result, the stain is not able to come out due to the impervious pore coats. Then we counter stain it with safranin. So under a microscope, it will appear green within red cells under the oil immersion lens of compound microscope. We require Bacillus cereus or Bacillus subtilis containing endospores culture. We require the staining reagents such as malachite green, safranin, and distilled water. What else? We require glass lights, clean grease-free glass lights, inoculating loop, spirit lamp, blotting paper, microscope. Now, what is the procedure? First, we take your smear, we prepare smear of culture on a clean grease-free slide. We air dry the smear and then we heat fix it. After the heat fixation is done, we stain it with malachite green, see, and keep it on a hot plate. Because of heating, it helps in the penetration of the primary stain. Then after keeping it for 5 minutes, we wash it with water. We cool it first and then wash it with water. And then stain it with safranin. And again we keep it for 1 to 2 minutes. And then we wash it with water. Air dry the smear and then we see it under the compound microscope. Those vegetative cells which do not contain any spores, they will take up the counter stain only. That is, they will appear red only. And those vegetative cells which contain endospores will appear green within red cells. Now, the position of the endospores helps in the identification of the genera of bacteria. So, in this way, through endospore staining, in which we use differential staining, we can identify the genera of bacterium which is pathogenic to human beings. The primary stain should not boil during the heating step. Air drying and heat fixation needs to be done with gentle care, and while washing it, the water should not be dropped to the center of the slide as it may cause a disruption in the smear. Capsules are extracellular material which is present outside the bacterial cell. It is generally present in certain pathogenic bacteria like Klebsiella pneumoniae. Now, what is the theory behind the capsule staining? We generally follow the Anthony staining procedure, in which we use crystal violet. Now, we know crystal violet is a basic stain. So, it will bind with the negative components of the bacterial cell. And what is the decolorizer used here? That is the copper sulfate. Copper sulfate in this case acts both as a decolorizer and a counter stain, because the capsule is rich in polysaccharides. As a result, it won't be able to bind with crystal violet and it will take up the color of copper sulfate when it is used as a decolorizer. So, at the end of staining procedure, you will see capsules as a light blue background or a light blue color within a deep purple color. So, the deep purple color is that of the bacterial cell, while the capsule has taken up a light blue color. We require cultures of Klebsiella pneumoniae, we require copper sulfate, we require crystal violet solution, Apart from this, we want clean grease-free slides, inoculating loop, wash bottle and microscope. We take a clean grease-free slide, a loop full of Klebsiella pneumoniae culture is taken, smear is made, the smear is allowed to air dry, and then we flood it with crystal violet and keep it for 2 minutes.
Ma'am, why is the smear not heat fixed in this case?
Because during heat fixation, some of the bacterial cell might shrink, and if it shrinks, then morphology will be altered and we won't be able to see the capsules carefully. So during capsule staining, the heat fixation step is omitted. Then after keeping it for 2 minutes, it is washed with copper sulfate, dried and visualized or seen under the microscope. We will see that the capsules, they have taken up the copper sulfate color and hence they will appear light blue in color, while the crystal violet, which is a basic dye, will enter the cytoplasm of the bacterial cell and it will become deep purple in color. So in this way, we can differentiate between the capsulated and non-capsulated group of bacteria. First is we will not heat fix the slide. Why? Because it might cause shrinkage of the bacterium. Then we won't be able to see the capsules easily. And the another important point is we do not use water, which we use in the other staining procedure for decolorization, because the capsules, they contain polysaccharides and polysaccharides is soluble in water and hence the bacterial cells will be washed away during the washing step. And we generally take a greater amount or a heavy smear of bacteria as we are not heat fixing, so some of the bacteria might be lost during the staining process. The type of differential staining procedure which we will follow in our microbiological lab depends upon the type and nature of bacteria. Whether it is gram positive or gram negative, accordingly we will follow. The second important point is the differential staining helps us in the identification, classification of microbial organisms, which is very important because based on this we can build the taxonomic characterization or we can build the taxonomic classification of bacteria. The entire staining procedure is based on the chemical properties of bacterial cell or its components. Thereby, it helps us to differentiate or to identify the medically important clinical samples which are responsible for the various diseases in human beings.